I'm Carrie Kodama. I'm a postdoc for the WRRC, and I'm the seminar co-coordinator with Aurora Kagawa Viviani, who I haven't seen come in yet, but she's been hard at work with the Water Commission and the Red Hill Task Force at UH. So she's been very busy in these last couple months. I'll let her introduce herself if she makes it in later. I'd also like to introduce the director of WRRC, Tom Jambaluka, who I'm sure many of you know, but Tom, if you want to say a few opening words before we turn it over.
Thanks, Carrie. Yeah, and thank you and Aurora for all the hard work you do to coordinate the seminar series. And thank you everyone who's tuned in for the first seminar of this semester. And I'm sure Carrie is going to mention this, that we intend to focus the semester's seminars on the Red Hill topic, not exclusively, but you know, we'll focus you know, as much as we can on this issue this semester. And so please be on the lookout for future seminar announcements as they come in. Thank you. I'll turn it back to Carrie. Thanks, Carrie.
Yep. Yeah, as Tom said, we're having a general theme of the Red Hill water crisis. And so we're trying to bring in various experts on impacts and mitigation efforts involved. So today we're starting off with one of our faculty members at WRRC, Dr. Tao Yan, who will be talking about the biodegradation of petroleum hydrocarbons. Dr. Tao Yan is a professor at the Department of Civil and Environmental Engineering and Water Resources Research Center at the University of Hawaii. His research interests are in the areas of environmental health, microbiology, and has recently focused on the biological quality of Hawaii's coastal water, wastewater infrastructure for further human health protection, including pandemic detection, bacterial antimicrobial resistance in natural and built environments, and biodegradation of recalcitrant organic compounds in the environment. He is a strong advocate and practitioner of the transdisciplinary education of the next generation of environmental engineers and has mentored numerous MS and PhD students and postdoctoral research fellows. Before I turn it over, just some quick logistical notes. The presentations in general will be between 45 and 50 minutes with time for questions at the end. So please hold all your questions until that time, or feel free to leave them in the chat so I can call on you later to either read your question or I can relay it to the speaker for you if you prefer. And you'll also see a periodic reminder in the chat to fill out an attendance form. Please do so, as this will help us keep track of our audience engagement. And with that, I'd like to turn it over to Dr. Yan.
All right, thanks, Carrie, for that introduction. So today I'm going to talk about biodegradation of petroleum hydrocarbon. And my title here actually is a little bit more than what I'm going to discuss, because as I was preparing this, I found that the material in this topic is tremendous. So I will today spend the time focus on the biodegradation part and talk a little bit about some case studies in terms of how biodegradation controls feed and transport in subsurface environment. Okay, so I want to give a quick introduction about my own experience with petroleum hydrocarbon and biodegradation. And I did that was way back 2000. I did my master degree in environmental engineering back in China. And I worked on this numeric modeling of groundwater flow and hydronic containment of petroleum contaminants in this city called Zibo City in Shandong province, China. And that's where the China has a whole bunch of several large government-owned petrochemical companies, and this Sinopec is one of the biggest. So, and this company called Qilu Petrochemical Company operates in Zibo City, and their petrochemicals, petroleum hydrocarbons, contaminated the groundwater. So my task then was to do some numeric modeling. And then I came to the United States, did my PhD in biodegradation. And at that time, the project was on polychlorinated biphenyls in sediments. So my focus was on how microorganisms under anaerobic conditions can remove chlorines from these polychlorinated compounds. So think about this, almost 20 years later, and then in Hawaii, we are also running into problems of petroleum contamination, and also, as biodegradation is one of the most important factors controlling the fate and transport of these pollutants in the environment. My experience 16 years ago seems will come back to be of useful. So I want to first talk about forever chemicals, because I worked on PCBs, and back then PCBs at the beginning in 1960s, 70s was considered to be a forever chemicals. And of course, now we know that PCBs can be degraded. But when you look at the chemical structures of PCB, which is a biphenyl ring, and with hydrogen substituted by chlorine, so this substitution can happen at any of these locations. That will give you a very complex mixture of PCBs. You can have up to 2009 congeners. And there was a very interesting story about how our perception of the biodegradability of PCBs have changed. At the beginning, everybody believed that it's going to be in the environment forever. But as we started to look at natural environments, then people started to realize that the chromatograph of PCBs in river sediments, in particular in Hudson River, where General Electric dumped a lot of these PCBs into Hudson River, the chromatograph actually shifted from the more chlorinated compounds towards the less chlorinated compounds, which indicates that there's dechlorination going on. That really started the process. So I spent 4 years on my PhD looking at how we can enhance the PCB anaerobic dechlorination. So this is one of the figures that I got in my PhD dissertation. I just wanted to look at the x-axis, which is time. So from the point I set up the reactor, it took 200 days for me to observe something happening in the system. So in this graph shows that the more chlorinated, more highly chlorinated biphenyl, which is 2, 3, 4, 5 chlorobiphenyl, will gradually lose chlorines, and you will start have this less chlorinated compounds. And my PhD dissertation led to this one paper that I'm pretty proud of, which is that we were the first group that identified that these microorganisms responsible for PCB anaerobic dechlorination are indeed this group of organisms called dihalococoids. And these are a group of obligatory dehalorespirers, meaning that they can only use chlorinated organics as their terminal electron acceptors for their respiration. So that was a paper that we published, and which later on, of course, there's more study that showed that corroborated our observation there. So by talking about PCBs being forever, but then we know that they're going to be degraded. I also want to mention that recently we are all probably heard of this another group of persistent organic compounds called PFAS, and this is also considered to be forever several years back. But now there's also evidence showing that microorganisms can degrade those chemicals as well. So when you think about these organic compounds, and even those highly fluorinated compounds can be biodegraded, then for petroleum hydrocarbon, so they are not going to be forever when they enter into our environment. And this is true, if you just think about how many oil spills that has occurred, both in water and on land. For example, I have this couple of major events of oil spill in ocean water, start from 1969 Santa Barbara oil spill to the most recent 2010 Deepwater Horizon spill. So a huge amount of oil gets spilled into the ocean water, which is shown on this NASA photo here. You can see this sheen of oil on the ocean water surface. So those petroleum hydrocarbons, some of them will end up in the marine sediments, but a lot of them will be dispersed into the ocean water and eventually be degraded. Another common occurrence of petroleum hydrocarbon contamination is oil spills on land. And this is most frequently linked to these underground storage tanks, USTs. So this, since the establishment of a UST program in 1988, the law actually was passed in 1984, and there has been documented more than 500,000 releases by these USTs. And even with all this stringent regulation, inspection, monitoring, today you can still expect about 5,000 new releases every year. And the most intimate example to us is this recent red hell spills. So I want to also, before I go further into biodegradation, I want to lay a little bit more foundation on what are petroleum hydrocarbons, which I'm sure a lot of you are very familiar with. And when you look at a petroleum hydrocarbon, it's really a very, very complex mixture. Just like the PCBs that I talked about, you can have 2009 different congeners. When it comes to petroleum hydrocarbon, you can have numerous, a large number of different types of hydrocarbon compounds. They can be, some of them, a very small fraction can be polar. Most of them are non-polar. And among the non-polar hydrocarbons, and we're very familiar with the alkenes, the cyclicals, alkenes, and aromatics. So I'm going to highlight here this mono aromatic hydrocarbons, MAH, because this group is the main risk drivers when it comes to petroleum hydrocarbon contamination. And this group includes benzene, toluene, ethyl benzene. So those are BTEs, and also the three isomers of xylenes, orthoxylene, methylxylene, and paraxylene. And these compounds, like I said, they are the main risk driver because they have one of them, benzene, is a known human carcinogen, and ethyl benzene is a possible human carcinogen. And when you look at the EPA's drinking water standards, and benzene has the smallest maximum contamination level, which is 0.005 ppm. So when later on, we're also going to talk about the solubility of these different petroleum hydrocarbon compounds. You will see that these aromatic compounds have the best water solubility. So when you combine a water solubility to their toxicity, then it's easy to understand why most of the study today on the feed, transport, toxicity, health impacts focus on BTEX compounds. And some study also include this MN to the end, methanaphthalene and naphthalene to it, because their solubility and toxicity are also high. So petroleum hydrocarbon can be degraded. So they are not forever chemicals. And when you look at how petroleum hydrocarbon are degraded in the environment, so this is a general chemical reaction that outlines the components here involved. So I use this 6HYOZ to indicate hydrocarbon. So here, of course, we shouldn't have that O there because these are hydrocarbons. And so this hydrocarbon, they function as electron donors. And in a redox process mediated by microorganisms, you should have electron acceptors. So this can be oxygen or can be non-oxygen electron acceptors like nitrate, sulfate, ferric iron, magnesium, so on and so forth. And then this process is mediated by microorganisms. Microbial biomass function as the catalyst in this reaction. And in that process, the microorganisms benefit and they grow their biomass or they maintain their biomass. And the carbon, the petroleum hydrocarbon gets oxidized in carbon dioxide and water. So when you think about this degradation process from this general chemical reactions, you can say that this is really a tripod. The reaction needs three footing. The first one is that you need to have these microorganisms. These microorganisms need to have the metabolic capabilities to carry out those biodegradation. So they can either be anabolism or catabolism. So either they just degrade the petroleum hydrocarbon for energy, catabolism, or they use the carbon to build in their biomass, anabolism. And that is also how these microorganisms build a biomass. So you need to have enough biomass to achieve fast enough degradation rates. The second second group of key components is the hydrocarbon themselves. And the petroleum hydrocarbons, they're not always bioavailable, and the different compounds can have different solubilities, but in general, they're not very soluble. They're generally hydrophobic. Therefore, their low solubility can restrain their availability to microorganisms for biodegradation. And even also among these different petroleum hydrocarbon compounds, some are more likely to be degraded and some are less likely to be degraded. For example, when you look at alkane versus BTEX-MN compounds, alkanes tends to be more easily degraded by microorganisms, whereas BTEX-MN compounds tends to be more resistant or recalcitrant. The third group of components is environmental conditions. So here we're looking at a biologic system where the microorganisms needs to grow. I talk about, there are microorganisms that will use oxygen as their terminal electron acceptors for their respiration. So those are aerobic microorganisms. There are also microorganisms that don't or can't use oxygen as their electron acceptor, and they rather use some other non-oxygen terminal electron acceptors, or TEAs, such as nitrate, sulfate, ferric iron. And those will also determine how fast, whether or not biodegradation will occur and how fast they occur if they do. Some other environmental conditions like nutrients availability, like nitrogen and phosphorus, can also affect the biodegradation process. So first, I want to talk about microorganisms that are capable of degrading petroleum hydrocarbons. So I want to start with this Supreme Court case. Well, I found this to be quite interesting. And back in 1980, there was this landmark US Supreme Court case decision, Diamond versus Chakrabarty. And Chakrabarty was working for General Electric. And he filed the first US patent for genetically modified organism. So he called it multiplasmid hydrocarbon degrading Pseudomonas. So hydrocarbon here is specifically for oil, petroleum hydrocarbon. So the US government rejected that. So they had a legal fight, and the case eventually ended up at the US Supreme Court, which determined that a live human-made microorganism is patentable subject matter. So basically it says that if you create an organism, then you can patent that and make money out of it. So this was the first ever US patent on a living organism. And it is really the start of the US biotech industry. So today, when you look at all these biotech companies, seed companies, monoclonal antibodies, so on and so forth. And it's really this landmark Supreme Court case that started the whole thing. But what is Pseudomonas? And this organism itself is actually very commonly found in soil bacteria, which also speaks to other bacteria that can degrade petroleum hydrocarbon, which is also very commonly found in environment and very diverse. So Pseudomonas is a group of common soil bacteria, and now we know that many Pseudomonas, not just Pseudomonas putida, which is the organism that that Supreme Court case was about. Many Pseudomonas can degrade diverse organic compounds, including petroleum hydrocarbons. And this Pseudomonas putida, this bacteria itself, has a very interesting story. So it was first isolated in Japan in 1960s. So this is the first organism. It was easy to isolate. So nowadays, if you go out try to isolate Pseudomonas from soil near on campus, you will be able to do that. And so this organism was isolated and then was shared widely with researchers around the world. And in 1973, people found that this strain, Pseudomonas putida MT2, was able to degrade aromatic compounds, hydrocarbons from oil. And then 1974, researchers identified that degradation capability was linked to this plasmid. Plasmid is a piece of DNA in the bacteria cell that can be moved around from one species to another, to one cell to another. So this was really the first catabolic plasmid that was identified. At the time, most of our understanding about plasmid was on antibiotic resistance plasmid. Then 1976, this plasmid was isolated. And then 1981, this first patent on a bacteria GMO was filed. So there was the original researcher who isolated Pseudomonas putida actually wrote a really nice long story about how this one microorganism from Japan traveled around the world and reshaped the landscape of microbial genetics. So what the contribution from Professor Chakrabarty is that he created a superbug. So what he did is that, before then, there are other researchers who isolated these two groups independently, identified this plasmid. It's called a TOL plasmid, PWWO. And these researchers found that this plasmid encodes metabolic capabilities that use toluene and xylene, which are two of the BTEX compounds that we're going to talk about later, as the sole carbon source for growth. And they also found that this plasmid can be transferred horizontally from one bacteria to another. That also has implication later on, because if you have plasmid that can degrade the petroleum hydrocompound that can transfer from one species to another, then you would expect this capability to be widespread in the environment. Some other researchers, so here, this is a biochemical pathway that shows how this tall plasmid in Pseudomonas putida were able to degrade toluene through these different pathways. So around that time, several other research groups have also identified other plasmids that are able to degrade other petroleum hydrocarbons, not only toluene and xylene. So what Chakrabarty did was he took those different plasmids, he put them into Pseudomonas putida, and then he fixed them in those cells with UV irradiation. So that led to this, he called Pseudomonas putida superbug, which demonstrated very high efficiency in degrading oil spills than any of the other individual bacteria species, Pseudomonas species. GE didn't really make a lot of money out of this case, because later on people found that they can just go out to the environment and isolate different types of bacteria with the capability of degrading petroleum hydrocarbons. So today, some reviews have indicated that there's more than 79 different bacteria genera that can degrade petroleum hydrocarbons. So I copied a small sublist of bacteria genera that are commonly found in the soil environment that can degrade petroleum hydrocarbon. So the ubiquitous distribution of these bacteria means a couple of things. So first of all, petroleum hydrocarbon in the environment will not stay there forever. So they will eventually be degraded, which also means that if you go out to the environment and you try to detect for petroleum degrading bacteria, that detection itself doesn't necessarily mean that significant occurrence of biodegradation, because you will always detect something out in the environment. And what is really important here is how much can you detect, because the biomass of those microorganisms will determine the reaction kinetics, which will determine the extent or rate of biodegradation of petroleum hydrocarbon. So I just want to give a quick review of some basic biodegradation kinetics. So when you look at how microorganisms degrade substrates in the environment, if the substrate is the rate limiting substrate, so in this case, BTEX or other petroleum hydrocompound, and its degradation rates can be expressed by this differential equation. So S stands for substrate. So how fast the substrate is changing is a function of K, which is the maximum specific substrate utilization rate. S, which is the substrate concentration. X here stands for the biomass divided by this Ks, which is half saturation coefficient plus substrate concentration itself. So when you look at how fast a substrate can degrade in the environment based on monod kinetics, here we assume that the bacteria is not growing. So this X here, this is the biomass, is not going to be changing for that short period of time. How fast the petroleum hydrocarbon can degrade depends on the types of microorganisms you have in the system, because that will determine K, sorry, not mu max, K, that's the maximum specific substrate utilization rate, as well as K sub S. So that is the half saturation coefficients, which indicates the affinity of that bacteria towards the substrate. And also the biomass of the microbes that degrades those petroleum hydrocarbon is also important. So that's this part. And also the limiting substrate concentration S. So this equation really speaks to that three components of biodegradation of petroleum hydrocarbon in the environment. Of course, monod kinetics is not the easiest model that people like to use. And later on, we're going to introduce some of the kinetic observation in the laboratory and also in the laboratory and also in field observations. And people tends to use first order reaction kinetics to describe it. And so if you take this monod kinetics and you focus that on this region where the substrate concentration is way less than the half saturation coefficients, K sub S here, then this monod kinetics can be simplified into this form. And then we can simplify this Kx divided by K sub S as K1. So that gives you a first order reaction kinetics approximation of monod kinetics. So talk about those, we have microorganisms that can degrade petroleum hydrocarbon. And another component is the hydrocarbon themselves. Different hydrocarbons have different properties that can affect biodegradation. So solubility is one of the most important parameter that affects the bioavailability and the degradation of those compounds by microorganisms. This is because microorganisms have to uptake those compounds into their body, or at least on their membrane, on their cell surface, before their enzymes can attack these compounds and degrade them. So when you look at the solubility of the different petroleum hydrocarbons, you can say that they're, based on whether you're talking about aliphatics or aromatics, you're looking at two different types of behavior. Generally, for the alkenes and alkenes, they have very low solubility. And when the carbon number increase, then the solubility drops precipitously. Aromatics, on the other hand, have higher solubility. So aromatics, these are the blue circle here, have higher solubility than the aliphatics. Note that this is a log scale. So, their solubility actually are a lot more higher than aliphatics. And they also follow the similar trends, more carbon, effective carbon number in those compounds, you will have less and less solubility. So one thing to note is, when you look at the aromatic compounds, the BTEX compounds, we said that BTEX compounds drives the health risks of petroleum hydrocarbon pollution in the environment. Benzene has the highest solubility, 1800 milligram per liter, toluene is 530, xenon, those three isomers have 160 to 190 milligram per liter. So these are a lot higher than the aliphatics compounds. So another factor that comes into play is that when you think about petroleum hydrocarbon mixture in the environment, it's a mixture, and how each of those compounds to dissolve in water actually has to fight between partition in the fuel mixture itself and to dissolve in water. So that partitioning can be calculated or can be indicated by this concept called effective solubility. So this is really, when you look at a LAPO mixture, non-aqueous phase liquid, its solubility is equal to its pure solubility of pure compounds times its molar fraction within that LAPO mixture. So that gives you this this SIE, effective solubility. Another factor that has a lot to do with bacteria for petroleum hydrocarbon is pseudo-solubility, and this actually can have implications in transport behavior of these compounds. So it turns out that there's a lot of microorganisms that can degrade petroleum hydrocarbon, these hydrophobic compounds. But these hydrophobic compounds, in particular alkenes, have very low solubility in water. So in order for those microorganisms to degrade them, they want to get these alkenes into water. And the way they do that is by secrete biosurfactants into the environment, and those biosurfactants will then go attach onto the oil droplets. Then they will create this emulsification. And typically these are small oil droplets between 0.1 to 1.0 micrometer size. And that's about the same size as microorganisms here. And then, that can significantly increase the surface area of the oil in the environment and allow bacteria to grow on those alkenes specifically. So this is a picture from a study from this group, and they showed that when they add bioemulsifiers, emulsifiers produced by bacteria into this oil water mixture, and they are able to create this water in oil emulsion on top, as well as this oil in water. The up is water in oil, the lower is oil in water emulsion. So basically, creates a lot of significant more surface area for the microorganisms to approach. Whereas when they don't have bioemulsifiers, they don't really see that phenomenon. So I also mentioned that different for the petroleum hydrocarbon, this mixture, different components, different type of petroleum hydrocarbon can have different biodegradability, and typically this general trend of degradability is observed. So linear alkanes tends to be most biodegradable. Then it's branched alkanes. Then it's monoaromatics, BTEX compounds. Then it's cyclic alkanes and the polycyclic aromatic hydrocarbon PAH compounds like naphthalene, like methanaphthalene, pyrene. Those are very, very resistant and recalcitrant in the environment. They're very resistant to biodegradation. So how fast can these compounds be degraded? So I looked up some review papers. This review paper reported this first order rate coefficients here. And this, they found that petroleum hydrocarbon under aerobic condition has a first order rate coefficient 0.445 day to the power -1. Whereas when you look at BTEX, benzene, ethane benzene, toluene, and xylene, their rates are much smaller. This is almost 1,000 times smaller than petroleum hydrocarbon. Of course, these numbers, these rate coefficients, are good for intra-study comparison, like within the same study, because remember that we talked about this first order rate coefficients. So this K1, it is a function of this K, that's the maximum substrate utilization rate, this Ks, K sub S, which is the half coefficients, but also depends on the biomass concentrations. For different biomass in a study, then you can have different rate coefficients. So the third group of environmental factors that affect biodegradation of petroleum hydrocarbon is the environmental conditions. The most important environmental condition here is oxygen or local oxygen. So we have studies have shown that when petroleum hydrocarbon are present in the environment with good availability of oxygen, then the aerobic biodegradation will occur, which is typically much faster than anaerobic biodegradation. And this, which is also why most of the laboratory and field observation experiments focused on aerobic biodegradation. But petroleum hydrocarbon will not only stay in aerobic condition. For example, when you look at the oil spill in the ocean, most of the oil eventually will end up at the ocean sediments. When you look at the land, fuel spill on land by USTs, and these oil, they may end up in the vadose zone, in the soil, in the vadose zone, which is supposed to be have air present, but the fluid themselves will occupy the space and then start biodegradation, aerobic biodegradation, consume those limited oxygen available in the vadose zone and create anaerobic environments. So when you look at the biodegradation of petroleum hydrocarbon in under anaerobic conditions, then researchers have found that many other non-oxygen terminal electron acceptors such as sulfate, nitrate, carbon dioxide, so these are methanogenesis, ferric iron, manganese, these alternative electron acceptors can also support anaerobic biodegradation of petroleum hydrocarbon. But when you compare that, the rate of degradation of anaerobic degradation to aerobic degradation. Anaerobic degradation is much, much slower. So another factor that's important here is, of course, temperature. And we all know that, for biological processes, you need to have optimum temperature to facilitate the growth of microorganisms to gain biomass so that they can function as catalysts to carry out the biochemical reactions. So here is a study done by Atlas et al. Back in 1975, and they showed that at 10 degrees Celsius and 20 degrees Celsius, they observed different rate of biodegradation in terms of evolution of carbon dioxide from their petroleum hydrocarbon. The third group of environmental condition that's important is nutrients and nitrogen, in particular nitrogen and phosphorus. So this is because when you think about microbial biomass, they are catalysts, but they need to grow themselves from very small amount to larger amount. And when you look at the microbial biomass, they need all these elements, and among them, nitrogen and phosphorus are typically limited in petroleum hydrocarbon contaminated environments. Some studies showed that to degrade 1 kilogram of petroleum hydrocarbon, you need at least around 150 gram of nitrogen and 30 gram of phosphorus. And this was also supported by studies in marine environments where when they add nitrogen and phosphorus to marine environments, they significantly increase their biodegradation rates, or in soil environments, like in groundwater aquifer, by adding nitrogen here. Nitrogen is more limited in terrestrial systems. Nitrogen amendments increase the biodegradation rates. So those are some of the, the three major areas of requirements, or three major groups of components for biodegradation of petroleum hydrocarbon in the environment. And I'm going to quickly go over some of one of the most studied petroleum hydrocarbon sites, and just talk about some of the observations that these scientists have made. And so this is at Bemidji in Minnesota. So that's the spill happened back in 1979. So this about 1.7 million liters of crude oil was spilled at the site. A good portion of them have been recovered through various means, but significant amount was left at the site. So this was the most studied site by USGS. Hundreds of papers have been published. Maybe not hundreds of papers. A lot of papers have been published. So what they have observed is that with all those efforts, they did 5 years of pump and skim, and then 30 years of natural attenuation. Back in 2009, they still observed a significant amount of oil at the subsurface. Another group of observation is that although spill happened in 1979, methanogenic degradation began by 1987, or observable methanogenic degradation of petroleum hydrocarbon was observed by 1987. And they also observed that substantial degradation of N-alkanes, those aliphatic, saturated aliphatic compounds, were degraded under methanogenic conditions, which was observed by looking at these chromatographs. So they also found that iron reduction was very important. The extracted iron, ferric iron in sediments near the oil plume, and which they found to be 30% less than the background, which means that the petroleum hydrocarbon has supported anaerobic iron degradation. And here, the ferric iron was used as the terminal electron acceptor, whereas the petroleum hydrocarbon was used as the electron donor. And they also observed organic acids are the main metabolic intermediates of biodegradation. and the biodegradation process is structurally dependent. This is basically they found that the persistent compounds benzene and ethylbenzene decreased slowly in the aloxic zone, whereas the more biodegradable compounds, toluene and xylene, decreased very quickly. And what's very most interesting to me was that they observed that anaerobic ion reduction limited plume growth. So when this was when they compared how fast the front of the BTEX was moving over time, and also they observed they monitored the different redox reactions from iron reduction to methanogenesis. They found that this iron reduction was controlling how fast the plume was growing. And they also did some further estimation of degradation rates. So for BTEX, 95% of the BTEX mixture was benzene, which corroborates the fact that benzene and ethylbenzene degrades really slowly at the site. And their estimation of the kinetics is 3 times 10 to the minus 3 day minus 1. So that's a first order reaction kinetics. To transform that into half-life, so that is equivalent of 231 days. So that's a quick summary of what are the major components of biodegradation, and some of the papers that I was able to I have been able to read at this well-studied site Bemidji. And now I would like to open the floor up for questions, comments, and hopefully we can have a discussion on this topic.
All right, thank you so much for the excellent presentation. So yeah, we're gonna open the floor for questions. We have 2 in the chat, one from Don Thomas and one from Paul Ayer. So I'll let Don go ahead first.
Okay. Actually, I got many questions, but I guess the first one is there a way to optimize the nutrient content that is required for your biomass to accelerate the breakdown of these products? And of course, where I'm going with this is, okay, we've got a spill site. Would this be an opportunity for us to actually do some research on that and look at ways of accelerating that breakdown process? Thanks.
Yeah, so that is theoretically possible. And actually at this Bemidji site, and they one of their study, they look at which location most biodegradation occurs. And they have found this site where the rainwater recharge occurs most. And that's where most of the biodegradation occurred. And their explanation was that because the rainwater carries in additional nitrogen, and mostly nitrogen and phosphorus, and that has led to the observed enhancement of biodegradation at the site. So it is theoretically possible to introduce nitrogen compounds to enhance the biodegradation, but you also need to be careful about the consequence, the inadvertent consequence of introducing nitrogen to a groundwater aquifer, because we also know that nitrogen compounds, especially nitrate in the groundwater, can also be important pollutants.
Okay, Paul, go ahead.
Thank you, and thank you, Dr. Tao Yan. I've been proposing that an oxidizing solution be injected into the subsurface beneath the tanks to break down the contamination there, which is a source of the contamination down in the aquifer. But we have to be careful not to that this process doesn't generate its own set of contaminants. So I wonder if there's a similar concern with your bacteria that you don't generate. I mean, is there a chance you could create a mutated toxic bacteria? Or are there other forms of contamination that you have to be careful with your process?
Yeah, good question. So, for biodegradation, when you look at the biodegradation process itself, it's mostly natural process. So you don't really do anything. They call it monitored natural attenuation. And if you want to implement bioremediation, so you want to intervene, then the option would be one, to provide nutrients like Don just mentioned. And the likelihood of that to create some more harmful bacteria down there is not very high. So of course you will have to conduct monitoring, and this can be done relatively easily, because if you are doing a bioremediation at the site, and then you will have monitoring wells, and you can look at, nowadays we have the capability to look at what are the microorganisms in the sample and how they are changing over time, and then you can easily delineate whether your action of promoting bioremediation at the site has led to the development of some hazardous or more toxic bacteria in the water.
Thank you.
Okay, we have time for at least one more question.
If I could just ask a very simple general question. Tao, first of all, thank you very much for that great talk. I learned a lot. But can you kind of outline how and what the limits would be for implementing enhancing biodegradation at Red Hill specifically? How would we go about it, and what are the limits, and what are the cautions that we should take in trying to enhance biodegradation?
Yeah, I think first of all, it's going to be costly. So that's no matter what you are trying to do. So we mentioned there are different processes. There's an aerobic process, which is a lot faster, and there's an anaerobic process, which depends on what type of alternative terminal electroacceptors you have there, like ferric, iron, manganese, or you pump additional non-oxygen electron acceptors like sulfate, nitrate in there, and all those processes are going to be costly. So that's number one. And number two is that you also need to be careful about the inadvertent consequences. If you're will that process actually led to the mobilization of these pollutants? Because when you look at the petroleum hydrocarbon, or JP5 in this case, and they are not very water soluble. So the most water soluble ones are BTEX compounds, and it seems like they're not very high in this particular blend. So when you promote bioremediation, then you could, because biodegradation products are mostly polar, so they're soluble. Bacteria is going to eat the food, and they're going to excrete some waste, that's some intermediates. So these compounds can travel even further and mingle with the water much easily and move much faster. So that's another potential factor that we need to consider.
Thank you.
But how will you introduce your bacteria to the contaminated system? It's all deep underground.
Oh, so you don't need to introduce anything. So when you look at the diversity of microorganisms capable of degrading petroleum hydrocarbon, and you can assume that they are there in the environment, and the limiting factor is how to grow them to a critical mass that can carry out biodegradation at a rate that's appreciable, that can be observed, or that's relevant to our lifespan. So it's really how fast those microorganisms can grow up. So that's why I talk about these environmental conditions, nutrients, nitrogen, phosphorus, because nitrogen and phosphorus are typically limited under those type of conditions. Temperature is another factor. If it's too cold, not here, but if you are in Minnesota, then the temperature can be really cold, really low in winter. So those factors, if you meet them, and then the biodegradation can speed up. Of course, there's also the fact that some of the compounds itself is toxic to microorganisms. So at the center of the plume, when you have high concentration of all those compounds, then the activity or the growth of microorganisms can also be limited. Then that will limit your reactive zone to the outer area sphere where the concentration and other environmental conditions are suitable for microbial activities.
Okay, we had one last question from Gina. She had a raised hand, and then that'll be it for, I'll let, if you have further questions, then we can send you Tao's contact information and you can follow up with him later on. Gina, go ahead.
My question is, have you considered how Japanese farming, Korean natural farming harvest the indigenous microbes, and they can quickly make a whole large amount? It just takes a little time and not much money by using, by harvesting them. Then, like you said, with the nutrients, they can make a large field of them and can make even a whole tank of them we are doing. So I wanted to ask you, they even have the core sample from under the Red Hill tank of the soaked cores. Is it possible to do a test where you attenuate the indigenous microbe with the JP8 eating microbes and see what the byproduct is? Because the 17 holes that originally were made, I asked the scientists that came to the second meeting for Red Hill in 2016 if they think it would work to make a slurry and pour it down the holes as like to chase them. But a risk may be, I don't know what the byproduct risk would be, but a risk would be the soil becomes so alive that it will become soft and the tank may sink down. But they said that's a good idea, but I wasn't supposed to talk about it there because the Navy said don't talk about it. A Navy PR person said don't talk about it, but I want to ask your opinion.
About injecting some bacteria, slurry of bacteria that you farm outside into underground, into the subsurface system? Is that
Korean natural farming and Japanese natural farming is not afraid of aerobic situation to make fertile soil, and the microbe will use the fuel or components as that's what they are hungry for, right? Those microbes. And if they are friends with the indigenous microbes, then it will it won't fight with each other when it goes down into that environment. I'm thinking. Yes.
So it sounds like you're talking about bioaugmentation, introducing some microbes from other sources into a system, environmental sources, and the chance of the success of that really depends on the consortium, the microbial consortium that you develop. Going back to the Pseudomonas putida that I talked about in the discussion, and that microorganism was so popular because when you grow them in the lab and put them into environment, they can still survive there. And a lot of other microorganisms can't do that. So I don't know you know, a lot of other microorganisms can do really well in the laboratory conditions, but once you put them into different environment conditions, and they die off rather quickly. So I don't know, you know, when you talk about augmenting the microbial system, whether those microbes will survive and thrive under those conditions needs to be investigated.
I understand what you're saying. I mean to say attenuate, which means to introduce the local microbe in the soil with the microbe that eats that fuel and make them friends so that they do survive with each other. You know, otherwise they fight and they disappear.
Yeah, that needs to be, I guess you can try to investigate that in the laboratories to see if they can survive in optimal laboratory conditions, and then try it out in an actual system.
I don't want to take up all the time. Thank you.
Okay. Tom requested we get in one more question. So sorry to keep people a bit longer, but Barry, would you like to go ahead and ask your question?
Well, the one I put in chat, is that what you're talking about?
Yeah.
Can the microbial degradation be spatially modeled if we had sufficient monitor wells to define where the plume is?
Hey, Barry, can you repeat that question? Sorry.
I'm wondering if you could spatially model the degradation rate if we knew where the plume is.
Yeah, if if we knew the size of the plume, that's one part, and another part is that we need to know the biomass concentration, you know, within that plume, and we also need to know what are the environmental conditions, like, you know, whether it's aerobic, anaerobic, does it have enough nutrients, nitrogen, phosphorus, to support those degradation. And so then we can try to model that process. And of course that has to be corroborated or calibrated by field observation, like in the groundwater, you know, what do you observe there?
Thank you.
All right. I think that'll wrap it up for today's seminar. Thank you everyone for joining us. And if you want to follow up with Dr. Yan, get his contact information, please feel free to contact me. I'll leave my email in the chat, and I'll put you in contact. Our next seminar will be 2 weeks from now on March 4th, and that will be given by Dr. Han Tseng, and she'll be talking about her dissertation research on cloud water interception. But yeah, thank you so much, Tao, for giving that talk and taking the time to discuss with us today. Thank you everyone for being here. And Tom, if you want to give any closing remarks.
Yeah, just to reiterate, thanks to our speaker, Tao, great job. And to you, Carrie and Aurora, and behind the scenes, April and Pat, for facilitating the seminar, and to everybody who tuned in. Please keep in touch and look for our future seminars, including the one that Carrie just mentioned on March 4th with Han Tseng. Thank you, everybody. Well, aloha.